Hey guys, it's Miss Ottolini. Again, I'm going to be discussing the basics of cell signaling notes, but I'm going to focus on the sections that talk about the types of cell-to-cell communication based on the distance between cells, and also I'm going to discuss the effects of a change in a signaling pathway. Okay, so first I'm going to discuss the types of cell-to-cell communication based on the distance between cells. So cells can either communicate when they are directly in contact with one another, they're physically connected. They can communicate over a short distance from one another, or they could communicate over a long distance. An example of cells that are directly contacting one another are plant cells that are squished up next to each other. Here you see the cell walls of adjacent plant cells. And they are connected by plasmodesmata, which are holes in the cell walls of adjacent plant cells. Now, they allow materials to pass from the cytoplasm of one plant cell into the cytoplasm of another. So I just drew a little picture to help me visualize what this was describing. Okay. Another example of cell communication by direct contact happens in the human immune system. So what takes place is if you have a pathogen in your body like a bacteria cell, for example, this unhappy face is our big bad bacterium, you have cells within your immune system called macrophages, which means big eater. So they will literally engulf the bacteria, break it apart, so you see the unhappy face, the bacteria being chopped apart, and then they will literally present a portion of that bacteria on a protein on the surface of the macrophage. One of my students back in the day likened this to someone, like a soldier, killing an intruder and presenting its head on a pike to show others that, hey, this is the bad guy, you got to kill these guys. So this kind of happens in the immune system when the macrophage, which is kind of like a general guard, kills the first intruder, presents the head, and says, hey, don't let anybody like this in. This guy's bad. So he's presenting a part of the bacteria, and another cell within the immune system, the helper T cell, has to directly bind to that portion of the bacterium that's being presented by the macrophage. And that, is going to initiate a response. The helper T cell is going to cause the creation of two things. One, antibodies, which are proteins that float around and bind to any of that specific bacterium that's in the bloodstream or other body fluids. The helper T cell is also going to create these killer T cells, and the killer T cells are used to destroy any cells within the body that have already been infected by this particular bacteria. So we've got the antibodies that are in the bloodstream and in the body fluids, and those are attacking any free-floating bacteria. And then we've got the killer T cells to eradicate any cells that have been infected by the bacteria. And this all happened because we had this direct contact between the macrophage and the helper T cell. Now the rest of your notes about this topic discuss communication over short distances and communication over long distances. I'm just going to talk about communication over short distances briefly, because I wanted you to label the picture just to help you understand it better. So in the picture, we see a single nerve cell right here, and it is connected to this nerve cell, and this nerve cell is also going to have this long rope that's extending off of its cell body, and that rope is called an axon. Okay, so here we see a single neuron, and it looks like it is releasing signal molecules at the end of this rope at these junctions called the axon terminals, or I guess they're like little buds. They're called the axon terminals or the axon tips, and they're going to release a signal molecule called a neurotransmitter into this area called the synapse or synaptic cleft. You really can't see the space between this nerve cell and this nerve cell right here, but you can in this picture. So I labeled this one, and I also labeled this one to indicate that they're the same thing. So we see the axon terminal from this first neuron releasing the neurotransmitter into the synapse, and this is a signal molecule. It is then going to bind to receptors on the second nerve cell right here. And those receptors are located on the tips of the cell body, shown in orange here, which are called the dendrites. And that's going to signal this nerve cell. Okay, so we've got the synapse or synaptic cleft, the space between the nerve cells. We've got the axon tips on our first neuron here. And this is a close-up of a single axon terminal, number 2. And then we've got the dendrites or receiving portion of the second neuron right here and also here. And this is, again, a super close-up. Okay. Now, I also wanted to talk about in this video the effect of changes in signaling pathways. And specifically, I want to talk about what happens when a person has type 1 or type 2 diabetes. Now, a normal pathway within the body that responds to high blood glucose after you've eaten a meal would be the pancreas releasing a hormone molecule called insulin. Insulin would normally bind to a receptor on the liver cell, and the liver cell would respond by opening this channel protein called GLUT. GLUT is going to allow glucose from the bloodstream to enter the liver cell and be stored there. So the response is ultimately lowering the blood glucose because all that glucose is getting stored in the liver cell. And that's going to prevent major fluctuations in blood glucose. Now, in type 1 diabetes, the pancreas is unable to produce insulin, so the liver cell never receives this signal, and we can't open the GLUT protein to remove glucose from the blood. So in that case, the blood glucose remains high. With type 2 diabetes, this typically occurs when a person has been producing a lot of insulin over a long period of time, and the receptor proteins on the liver cell are no longer sensitive to that insulin. So even though the insulin is created by the pancreas, it doesn't cause a response in the liver cell because the receptor's like, I've seen too much of you before, I'm not going to trigger a response. So again, the channel protein GLUT doesn't open and the glucose does not get transported into the liver cell, so it's the same effect. The blood glucose remains high. All right, that is it. Thank you.